by which of the following statements? It produces thyroid hormones. These thyroid hormones are produced by tyrosine. So tyrosine produces the thyroid hormones. T3, T4, that is triiodothyronine, tetraiodothyronine. Each of them is made of 2 molecules of tyrosine. So the thyroid hormones, they come from tyrosine. They are not coming from tryptophan. Option B, it produces pineal hormone melatonin. Yes, tryptophan produces the melatonin. This is correct. Melatonin comes from serotonin pathway only. First, we will form serotonin, which will then be converted to melatonin. So yes, tryptophan forms melatonin. Tryptophan is required for the synthesis of 3 things. Number 1 is we will require tryptophan for serotonin synthesis. We require for melatonin synthesis. And we also require tryptophan for niacin formation. So niacin is vitamin B3, and hence niacin is not known as true vitamin because it can be synthesized endogenously from tryptophan. Rather, we will call it a endogenous vitamin because tryptophan can form niacin. It produces the pigment melanin. Melanin also comes from tyrosine. So, and even it produces catecholamines like dopa, dopamine, norepinephrine, epinephrine. That also comes from tyrosine. So, there are 3 things which are formed by tryptophan: serotonin, melatonin and niacin. In the given question, they are asking whether you know that what are the things which are produced by tyrosine and what are the things which are produced by tryptophan. And most of the students confuse between melatonin and melanin. Melanin is the pigment that gives us our color. Melatonin is responsible for the circadian rhythm, right? So question number 1, answer is it produces pineal hormone melatonin. Question number 2, all of the following amino acids are optically active except. Now, what is an amino acid? What do we mean by an amino acid? An amino acid is something which has a central alpha carbon which has 4 hands. One of the hand of the carbon atom is occupied by an amino group. Another hand is occupied by carboxylic group. Such an compound where a central carbon is occupying amino group and acidic group is known as amino acid. So this here is an amino group, this here is an acidic group, so it is known as amino acids. The 4th valency and the 3rd valencies are different. This here is hydrogen for all the amino acid, that is fixed. But the 4th valency, that is the R group, is different for all the amino acids, and depending upon what this R group is. If this R group is hydrogen, it will become glycine. So if I replace this R group with hydrogen, now one hydrogen is part of that amino acids, but another hydrogen is its R group. Glycine is the only amino acid. Glycine is the only amino acid where you will have the R group as hydrogen. There is no other amino acid which has R group as hydrogen. So all the following amino acids are optically active except glycine, because glycine is a symmetrical molecule. It is having 2 of its substituents are hydrogen, hence it is having symmetry. Now because of its symmetry, it is optically inactive. In other words, we can also call it achiral. Chirality means all the 4 hands of the carbon are different. All the 4 valencies of the carbon are substituted differently. But in case of glycine, this here glycine is the only one which is having symmetry, and hence it is the only amino acid which is optically inactive. So yes, it is glycine. It is glycine. Then all the other groups, serine will have hydroxyl group in it, CH2OH as the R group. The threonine will also have hydroxyl group. These are hydroxyl group containing amino acids. Both of them are hydroxyl group containing amino acids. But their R group will be different. Tryptophan has indole group containing amino acid. You must know the R group. What is the R group for all the amino acids? For example, by and large, we all remember that the two sulfur containing amino acids are cysteine and methionine. That means their R group is having sulfur in it. Both cysteine and methionine, their R group is having sulfur in it. Then question number 3. Which amino acid is represented by one of the stop codons UGA? Now, UGA, see what actually happened in the discovery of amino acids was, we were having 64 codons that were discovered. And we found out that 64 codons there are 3 stop codons and rest we have 61 codons. Now these 61 codons are coding only for 20 amino acids. They are coding only for 20 amino acids. That is why you will find that an amino acid can be coded by more than 1 codons. There can be more than 1 codon coding for the same amino acid. Now, which amino acid is represented by one of these stop codons? These 61 codons are fixed. For example, we know that AUG will always code for methionine, right? AGA, AGG will always code for arginine. AAA codes for lysine. Likewise, we have 61 codons. All of them are coding for different amino acids. Now, there are 3 stop codons. And these 3 stop codons are UGA, UAG and UAA. UGA, UAG, UAA. Now, after the discovery of the 21st codon, 21st amino acid, that is silanocysteine, the problem came that all the 64 codons have been assigned their functions. You know which codon codes for which amino acid. Now, what will be the codon for silanocysteine? That was the problem. Now, what they found out was that out of the 3 stop codons, UGA codes for the 21st amino acid silanocysteine. This was found out later because they have found out for the 20 amino acids, 61 codons were already assigned. So now it was very difficult for us to know what is the amino acid which is coding for UG for silanocysteine. So they found out that probably there can be a stop codon which for most of the mRNAs it will behave as stop codon. But when it has to form a protein which has silanocysteine in it, UGA does not behave as a stop codon. Rather, it binds to a specific tRNA which binds to silanocysteine. So here the answer is silanocysteine. So which amino acid is represented by one of these stop codons? The answer is silanocysteine. Then question number 4. Glutamic acid carries an extra carboxylic acid residue in the side chain. Hence, what can we say about glutamic acid? Let's see one by one. It cannot exist as Zwitter ion. Now see, Zwitter ion means the amount of negative charges and the positive charges are equal on an amino acid. If it is having a positive and a negative charge, it will be equated somewhere. So it will exist as Zwitter ion. It will carry a negative charge. Now here, this statement is partially true. In this given question, instead of mentioning it directly that it will carry a net negative charge, instead of this, they would have mentioned that at physiological pH, then this statement is true. So this statement is partially true. At physiological pH, then this statement is true. It will carry a negative charge at physiological pH. Yes, because glutamic acid is a type of acidic amino acid. That means its R group will contain a negative charge to it. So R group will have negative charge to it. Now its isoelectric pH will be lower than that of glycine. Now this is true. See, at any given point, whatever is the charge on glycine, the glutamic acid, being an acidic amino acid, will always have a lower isoelectric pH than that of glycine. So yes, this statement is correct. This statement is correct. This statement will be correct. So question number 4, answer is C. Question number 5. Catabolism of which of the following amino acid in the cell does not result in the formation of acetyl-CoA? So let's see. Now actually what happens with alanine is forming pyruvate. We know why a reaction known as alanine transaminase, which requires vitamin B6, and there is the enzyme ALT, you will form pyruvate. And pyruvate can result in the formation of acetyl-CoA by the enzyme. You all know this enzyme. Yes, which enzyme will result in the conversion of pyruvate to acetyl-CoA? It is by the enzyme pyruvate dehydrogenase. Yes, one of the 3 oxidative decarboxylation reactions. Now this alanine will first form pyruvate and then pyruvate will form acetyl-CoA. Now therefore, yes, we can say that yes, acetyl-CoA is formed from alanine. Now glycine also undergoes a transamination reaction. It also undergoes a transamination reaction where it will form pyruvate. And yes, pyruvate we just saw can form acetyl-CoA. So glycine can also form acetyl-CoA. Leucine is the only amino acid which is ketogenic in nature. Now because leucine is ketogenic in nature, it will form ketone bodies, and the ketone body is acetoacetate. To the acetoacetate, we will add acetyl-CoA. We will add CoA so that it becomes acetoacetyl-CoA. So that it becomes acetyl-CoA. Now this is having 4 carbon molecules. Acetoacetyl-CoA is having 4 carbon molecules. Now this acetoacetyl-CoA is then converted to 2 molecules of acetyl-CoA. For this you simply need a lyase, right? A thiolase, which will split that 4 carbon to 2 carbon, and you will get acetyl-CoA, which will enter into TCA. Now this aspartate is thus the only amino acid here which will not form acetyl-CoA. Rather, it is converted to oxaloacetate by the enzyme aspartate transaminase, and this is converted to, it can enter into gluconeogenesis, it can enter into TCA, depending upon which tissue, what is the status of our body, depending upon that this will occur. Now this way you can always say that catabolism of which of the following amino acids can be formed, and there are 20 amino acids. How to remember all these 20 amino acids? What are they forming? Where are they entering into TCA? For this is from basis of this question comes from amphibolic nature of TCA. So in the notebooks you will find there is a subheading amphibolic nature of TCA that must be discussed so that you come to know what are the various amino acids, where are they entering at the level of the TCA. That way you will come to know, okay, then these are the amino acids which are forming the pyruvate, which can form acetyl-CoA, these are the amino acids which can directly form acetyl-CoA and other intermediates. So to answer such kind of questions, you must have read amphibolic nature of TCA. And this topic becomes clear only after we have covered all the three metabolisms: protein metabolism, carbohydrate metabolism and lipid metabolism. Question number 6. The nitrogen in urea originates from. This is a very important question. Most of the students, they are confused with this. They always think ammonia is the source of the urea. Yes, ammonia is the source, but when you see the structure of urea, you will find that urea is having two nitrogens. One of these nitrogen is incorporated from ammonia during the very first reaction of urea cycle, where carbon dioxide reacts with ammonia to form carbamoyl phosphate. So this comes from ammonia. And the second nitrogen comes by the incorporation of the third enzyme, that is argininosuccinate synthetase, which is incorporating aspartate. So the second nitrogen, these have been found by the radio labeled assays. They have found this, that where is the first nitrogen, how is it getting incorporated, how is the second nitrogen getting incorporated. This have been serially, these have been studied over the years, where we have found that the first nitrogen is incorporated by ammonia and the second nitrogen is incorporated by the aspartate. But in the given options, now if you see, there is no option which is matching this. Now what to do? Right? Nitrogen in the urea originates from. Is there any option where you can see both ammonia and aspartate given? I can't see that. So now what we can do is, we can further see what is the source of this ammonia. The source of this ammonia comes from glutamine. And the source of this ammonia comes from glutamine. The glutamine in the liver is first converted to glutamate, which will then form alpha ketoglutarate, and then it will form release ammonia. So during both these steps, when glutamine is getting converted to glutamate, there is release of ammonia. And when glutamate is converted to alpha ketoglutarate, there is release of ammonia, and both these ammonia will enter to form the urea cycle. It will react with carbon dioxide, the first step of urea cycle. So in the given options, there is option 1 which is saying glutamate and aspartate. This is a trickier question, and hence my answer is glutamate and aspartate. Glutamate and aspartate, because the second nitrogen is coming from aspartate only, but the first nitrogen is coming from ammonia. The source of that ammonia is 2, glutamate as well as glutamine. Now have you understood this question? Question number 6, the answer is glutamate and aspartate. Question number 7. Which of the following is the coenzyme for transamination reactions? The answer is pyridoxal phosphate. This is a very important reaction. Most commonly, we take the example of ALT, AST, but there are more than 10-12 transamination reactions which are occurring at a given point of time. So, all the amino acids, they undergo transamination except 4. We will discuss them in a future question. For now, let's focus on how transamination occurs. Now, alanine contains an amino group. If you remove the amino group from alanine, structurally it is nothing but pyruvate. So, you have an amino acid on one side. You remove its amino group, it becomes a keto acid. So, this one here is a keto acid. But at the same time, this ammonia which is getting removed will not be going into the medium. It will combine with pyridoxal phosphate. It will combine with vitamin B6, and during the reaction, simultaneously, alpha ketoglutarate, which is a keto acid, is converted to glutamate. So, there is a transfer of ammonia. So, how this ammonia came? It jumped from here to vitamin B6, and then from vitamin B6 to ultimately it will land over to form glutamate. So, this ammonia is jumping from alanine to vitamin B6 and then to glutamate. So, during a transamination reaction, there is requirement or must need of vitamin B6. All such reactions are reversible. All such reactions are reversible, and there is no release of free ammonia. Free ammonia is not released here. Free ammonia is not released here. There is transfer of ammonia, hence the enzyme is called transaminases. Question number 7, answer is pyridoxal phosphate. Question number 8. Which of the following urea cycle enzyme defects are autosomal recessive except? There are 5 enzymes of urea cycle: carbamoyl phosphate synthetase 1, ornithine transcarbamylase, we have argininosuccinate synthetase, argininosuccinate lyase, and arginase. These are the 5 enzymes. Deficiency of any of these enzymes will result in high ammonia levels. These enzyme defects are called urea cycle disorders. All of them are autosomal recessive except ornithine transcarbamylase defect. This disorder is X-linked disorder. This is the only urea cycle disorder which is X-linked in nature, and it is this disorder which is also known as type 2 hyperammonemia. It is the second enzyme in the series of urea cycle disorders, so we call it type 2 hyperammonemia. Type 2 hyperammonemia. Question number 9. Which of the following is non-transaminable? Those which have lost the property of transamination. The mnemonic is lost. L-lysine, O-hydroxyproline, S-serine, T-threonine. These are the 4 amino acids which have lost the power to transaminate. I have purposefully added OH group here so that it will fit into the mnemonic lost. And as can be seen, there are 2 correct answers here. So there are 2 correct answers. Both serine and threonine will not undergo transamination. The rest, all the amino acids, they undergo transamination, and hence I always say that there are more than one transaminases. Just don't focus on ALT, AST. there are many other transamination reactions. There are tyrosine transaminase, the 3 branch chain amino acids, they have their individual transaminases. So these are the 4 amino acids which do not undergo transamination. Question number 10. The primary hyperoxaluria can be caused by the defect in the metabolism of. It is, see, like I told you, there are more than one transamination reactions. Similarly, when alanine is converted to pyruvate by ALT, there is another reaction which converts simultaneously glyoxylate to glycine. So here also you are having one amino acid which reacts with one keto acid to form another keto acid and to form another amino acid. So alanine will be converted to pyruvate, glyoxylate will be converted to glycine. This enzyme here is AGLT. This is also reversible. AGLT stands for A for alanine, GL for glyoxylate, T for transaminase. So there are more than 10-12 transamination reactions and AGLT is one of them. AGLT. A for alanine, GL for glyoxylate, T for transamination reaction, and this will require a vitamin B6. Yes, this will require vitamin B6. All transamination reactions require vitamin B6. But if there is a mutation of this enzyme, what will happen is there will be increase in glyoxylate. When glyoxylate accumulates, it will form oxalic acid. And this is associated with primary hyperoxaluria, and that will result in the primary hyperoxaluria is seen with glycine metabolic defect. It is seen with glycine metabolic defect. Then question number 10, answer is glycine. Question number 11. Which 2 amino acids serves as a precursor for creatinine? Always remember, creatinine is formed by 3 amino acids. I always use the mnemonic game. G for glycine, A for arginine, and M for methionine. Now, creatinine is a waste product. Its precursor is creatine. Whenever the half-life of creatine is over, it is spontaneously converted to creatinine. So, creatinine is more famous one because of its function that it is used for kidney function test. So, glycine, arginine and methionine. These are the 3 amino acids required for the synthesis of creatinine. So, here the answer is glycine and arginine. No other options are matching. So, the correct answer is glycine and arginine. 3 of the amino acids out of which 2 are there in the given options. Question number 11, answer is glycine and arginine. Question number 12. Maple syrup urine disease is caused by deficiency of the enzyme. This all of you know. The branch chain keto acid dehydrogenase. It is due to the deficiency of the enzyme branch chain keto acid dehydrogenase. This enzyme is further made of E1, E2, E3, where E1 is branch chain keto acid dehydrogenase, E2 is the dihydrolipoic acid, lipoic acid, dehydrogenase, and there is a dihydrolipoyl transacetylase. So there is a dihydrolipoyl transacetylase. So these are transacylase. These are the three E1, E2, E3 are together making one enzyme, and these are requiring 5 coenzymes. These include thymine, lipoic acid, coenzyme A, FAD and NAD. We have seen this earlier that there is one more reaction which is made of E1, E2, E3 and requires 5 coenzymes: PDH, alpha ketoglutarate dehydrogenase, and the third and last is branch chain ketoacid dehydrogenase. These are the 3 enzymes which are made of E1, E2, E3 requiring 5 coenzymes. This is important. You must remember what are the 5 things they require. Easy way to remember this is using a mnemonic that says tender, love, care for Nancy. Thymine, lipoic acid, coenzyme A, FAD, NAD. These are the 5 things required by this enzyme. Now I know you will remember tender, love, care for Nancy. You will forget what is tender, love and care standing for, but you will remember the mnemonic. Question number 13. In which condition urinary excretion of para-hydroxyphenylacetic acid is not seen? It is not seen. So let's see. See, during the para-hydroxyphenylacetic acid, it is formed during the tyrosine catabolism. In the very first reaction, tyrosine undergoes a transamination reaction and forms para-hydroxyphenylacetic acid. In the next reaction, it will form homogeneticic acid. This is very famous, you all know it. In the next step, we will form mallyl acetoacetate, which is converted to fumaryl acetoacetate, and in the end you will get fumarate and acetoacetate. In the end you will get fumarate and acetoacetate. Now, there can be a future question related to tyrosine catabolism. So I am discussing tyrosine catabolism in detail here. The tyrosine is converted to para-hydroxyphenyl pyruvate acetic acid by the enzyme tyrosine transaminase. The transaminases require vitamin B6. We already saw this in one of the previous questions. The para-hydroxyphenyl pyruvate acetic acid is converted to homogeneticic acid by an enzyme para-hydroxyphenyl pyruvate acetate, and the enzyme is hydroxylase. This hydroxylase requires vitamin C. The homogeneticic acid is converted to mallyl acetoacetate by the enzyme homogeneticic acid oxidase. Mallyl acetoacetate to fumaryl acetoacetate is an isomerase. Not a very important enzyme, I am not even writing it. But fumaryl acetoacetate is converted to fumarate and acetoacetate by the enzyme fumaryl acetoacetate hydrolase. Please note, if the last enzyme is deficient, it will result in tyrosinemia type 1. If the first enzyme is deficient, it is tyrosinemia type 2. If the hydroxylase is deficient, the second enzyme, then it is tyrosinemia type 3, which is also known as neonatal tyrosinosis or neonatal tyrosinemia. So during this here, This will result in high accumulation of para-hydroxyphenyl pyruvic acid. Now, if somebody is having deficiency of homogeneticic acid, this will result in L-captonuria. In L-captonuria, there will be increase in homogeneticic acid, and due to back pressure, there will be increase in para-hydroxyphenyl pyruvate also. So, L-captonuria will also result in high para-hydroxyphenyl pyruvate. Phenylketonuria is defect where tyrosine is formed from phenylalanine by the enzyme phenylalanine alpha hydroxylase. Phenylalanine alpha hydroxylase. If this enzyme is defective, then phenylalanine will be very high. So, phenylketonuria will result in high phenylalanine, low tyrosine, and hence low para-hydroxyphenyl pyruvate. Now, on the other hand, if somebody is not having vitamin C, still there will be high para-hydroxyphenyl pyruvate acetic acid. Why? Because this enzyme requires vitamin C. So, all these are causes of high para-hydroxyphenyl pyruvate except phenylketonuria, which is your answer. So, point number 1, you have to know tyrosine catabolism. Point number 2, you must know phenylalanine is an essential amino acid which gives rise to non-essential amino acid tyrosine. If the enzyme phenylalanine alpha hydroxylase is deficient, there will be accumulation of phenylalanine, and this disorder is phenylketonuria. Question number 14. Which of the following does not use pyridoxal phosphate as coenzyme? Now here, all the transaminases, they use vitamin B6. Glycogen phosphorylase is the rate limiting enzyme for glycogenolysis, that also uses vitamin B6. Kynureninase is the basis of the kynurenine metabolic pathway. Tryptophan via series of reactions is converted to first kynurenine, which by the enzyme kynureninase is then converted to alanine and acetoacetate. Alanine and acetoacetate. If somebody does not have the enzyme kynureninase, which requires vitamin B6, this is a very famous test known as tryptophan load test. In this, we will give large concentration of tryptophan. It is converted to kynurenine, which is then converted to alanine and acetoacetate. So, if the enzyme kynureninase has adequate vitamin B6, it will function. But if vitamin B6 is deficient, then kynurenine will accumulate and it is converted to xanthurenic acid. Xanthurenic acid. There is no source of xanthurenic acid in our body. It can only come from the kynurenine. So, this is known as tryptophan load test, and yes, enzyme kynureninase requires vitamin B6. Now, during the kynurenine pathway, there is another enzyme formylase which does not need vitamin B6, and hence the answer is formylase. Now, this you can tell. What is the more metabolic defect of tyrosinemia type 1? Fumaryl acetoacetate hydrolase. It's a poor prognosis disorder. In this, the infant usually dies because of hepatocellular carcinoma. Tyrosinemia type 1 is a lethal disorder. It usually causes hepatocellular carcinoma and death. So, question number 15, answer is A. Question number 16. Which of the following is known as Richner-Hanhart syndrome? Tyrosinemia type 2 is also known as Richner-Hanhart syndrome. So, answer is tyrosinemia type 2. Which enzyme defect? I already told you this in a previous question. Yes, Richner-Hanhart syndrome. It is due to which enzyme defect? Answer is the first enzyme, tyrosine transaminase defect. Question number 17. The neurological symptoms of phenylketonuria are due to reduced availability of tyrosine for protein formation. Tyrosine, of course, is reduced. The statement is true but is not a correct reason for the given assertion. Reduced availability of tyrosine for its neurotransmitter synthesis. Tyrosine forms dopa, dopamine and all that will result in neurological symptoms associated with phenylketonuria. If somebody is having phenylketonuria, phenylalanine cannot be converted to tyrosine. That means no dopa, no dopamine, no further formation of any catecholamines. Hence, there will be neurological symptoms. Also, high circulating phenylalanine can cross blood-brain barrier. It itself is toxic to brain. Decreased synthesis of tryptophan. Tryptophan is an essential amino acid. Phenylalanine forms tyrosine, not tryptophan. This also because of that, I don't know, because both tyrosine and tryptophan are starting with T. Many students have this confusion whether it forms tyrosine or tryptophan. Always note, phenylalanine will form tyrosine. Tryptophan is an essential amino acid. Both phenylalanine and tryptophan is essential. Question number 18. It is very easy for you now. We just discussed. Kynurenine is formed during metabolism of tryptophan. Aromatic amino acids are way more important than all other amino acids for any examination. You must know the two sulfur containing amino acids, cysteine, methionine, their metabolic pathways completely. The three aromatic amino acids, phenylalanine, tyrosine, tryptophan. In addition, the glycine can be asked. And at times you get a question on maple syrup urine disease that we have already discussed. So protein metabolism, these are the most important topics, and of course urea cycle. But that is the nitrogen skeleton. So from the nitrogen skeleton, urea cycle. From carbon skeleton, the specialized products of glycine, tyrosine, tryptophan. Whatever tyrosine forms is formed by phenylalanine also. Cysteine, methionine and the three branch chain amino acids, particularly the disease maple syrup urine disease. Question number 19. Which of the following is unique to collagen fibers? Collagen is a triple helix. You have 3 helices. Each of this helix is made of glycine XY like that. That means we can say that every third molecule in collagen is glycine. In other words, 33% of collagen is glycine. But at several places, proline is present. And wherever proline is present, it is converted to hydroxyproline. For this, we need an enzyme, prolyl hydroxylase. For this, we need an enzyme, prolyl hydroxylase, and this enzyme simultaneously will require vitamin C and iron. It depends upon two things, vitamin C and iron. Now what happens is, jahan proline hai, suppose there is a proline here, there is a proline here, proline here, proline here, proline here, proline here. Wherever proline is present, after the whole protein has been formed, as a post-translational modification, hydroxyl group will be added here. Once we have added hydroxyl group here, now what will happen is, because there are so many hydroxyl groups, now this will form hydrogen bonds. This will form hydrogen bonds wherever it is present. That is responsible for the strength of collagen. The hydrogen bonds is responsible for the strength of collagen. Now, let's say vitamin C is deficient. Now, no hydroxylation, no hydrogen bonds. and therefore this will result in poor collagen. Therefore, it will result in poor collagen formation. And this collagen is weak, that is why vitamin C deficiency is associated with scurvy. This is another very important post-translational modification. So, the unique to collagen fibrils is hydroxyproline. Question number 20. Deficiency of which of the following may be seen in megaloblastic anemia? Which amino acid? It is methionine. Methionine, via series of reactions, will first form homocysteine. The only difference between methionine and homocysteine is in the methyl group. So, if I have to convert homocysteine back to methionine, I only need methyl group. The donor of that methyl group is fixed, and that is methyl B12. This enzyme is a methyl transferase. I'll call it homocysteine methyl transferase. Now, let's say what happens here is, if somebody does not have if somebody is not having the enzyme homocysteine methyl transferase, there will be accumulation of homocysteine. If somebody is not having methyl B12, still there will be accumulation of homocysteine. But what is not getting formed? It is methionine. So, even methionine is requiring vitamin B12. And please note here, methionine is in the form of methyl donor. It is the universal methyl donor which will convert uracil to thymine. The only difference between uracil and thymine is of a methyl group, and that methyl group comes from methionine. Now let's say there is no methionine, that means no uracil, no thymine conversion. Uracil will increase, thymine will be less. That means there will be less of DNA, and hence because the DNA synthesis is retarded, there will be resulting in megaloblastic anemia. So yes, B12 deficiency results in the megaloblastic anemia. Also note that this B12 got this methyl group from N5, N10 methylene tetrahydrofolate. Of course, via one more step, N5 methyl tetrahydrofolate is the final donor. But yes, the central folate molecule is N5-N10 methylene tetrahydrofolate. That means even if somebody is not having tetrahydrofolate, still it will result in no methyl group coming from tetrahydrofolate, no methyl B12. There will be no methionine and no conversion of uracil to thymine. Uracil will increase, thymine will decrease, and hence B12, B9 and methionine deficiency all can be seen as megaloblastic anemia. Question number 21. Serine can be a precursor of each of the following except. Now, if I ask you, even if you do not know what all serine forms, there is a chance that you do not know what all serine forms. But even then, you know that methionine is an essential amino acid. Hence, methionine cannot be formed from serine. Methionine cannot be formed from serine. How it forms choline, how it forms ethanolamine, irrespective of that, this question is based on a simple basis that whether you know that there are 10 essential amino acids. I always use this mnemonic to remember 10 essential amino acids. You can use any mnemonic to remember 10 essential amino acids. P, phenylalanine, V, valine, 1T is for tryptophan, another T is for threonine, I, isoleucine, M, methionine, H, histidine, A, arginine, L-leucine, and another L for lysine. These are the 10 essential amino acids, out of which these two here are semi-essential. Histidine and arginine are required only in children. Adults can form, and hence they are called semi-essential amino acids. So question number 22. Homocystinuria can be all of the following except. We just said that methionine via series of reactions will first form homocysteine. Homocysteine is converted to methionine by methyl B12, and we also require here, we also need here enzyme homocysteine methyl transferase. At the same time, there is one more fate of homocysteine. It is converted to cystathionine by the enzyme cystathionine beta synthase. It is converted to the cystathionine by the enzyme cystathionine beta synthase. Now please note that this enzyme requires vitamin B6. Now if we see this carefully, deficiency of B6 will result in high homocysteine. Deficiency of cystathionine beta synthase will result in high homocysteine. B12 deficiency will result in high homocysteine. Homocysteine methyl transferase deficiency will result in high homocysteine. So let's see in the options one by one. Is it deficiency of homocysteine methyl transferase? Yes. Is it deficiency of cystathionine synthase deficiency? Yes. Is it malabsorption of vitamin B12 that it is not getting absorbed? Yes. Deficiency of tyrosinase is not at all related to homocystinuria. So question number 22, answer is C. Question number 23. What is the primary cause of cystinuria? It is a renal tubular transporter defect in which 4 amino acids are lost in urine. In the tubules, there is a transporter for the reabsorption of cysteine, ornithine, lysine, arginine. I use a mnemonic to remember this, and probably you have guessed it by now. So the answer is, what is the mnemonic? Yes, cola, whatever you want to remember, but it is the primary cause of the cystinuria is the renal tubular transporter defect in which the 4 amino acids are lost in urine, because of which that can also present with the cystine stones. It can present with cystine stones. So cystinuria can present, cystinuria also you can call it, but it is a renal tubular transporter defect in which 4 amino acids are lost in urine. They are not reabsorbed, lost in urine. Which of the following is oxidative deamination? The enzyme is glutamate dehydrogenase. Please note, we discussed this in one of the questions that glutamine is converted to glutamate, which is then further converted to alpha ketoglutarate. Both these reactions occur by release of free ammonia, but this free ammonia is not lost in the blood. Rather, it will be entering into the urea cycle. The next is urea cycle. So both these steps occur in liver. The first enzyme here is glutaminase, which will remove glutamine, say ammonia, and the next enzyme is glutamate dehydrogenase. During this step, glutamate dehydrogenase is present only and only in liver mitochondria, and this last step is known as oxidative deamination. Only this last step, and hence starting from transamination to this oxidative deamination, all of it together is known as trans-deamination. At times, you get a short note on what is trans-deamination. So it starts from transamination and it ends with oxidative deamination. Together, the whole of inter-organ exchange of ammonia, transamination will form all the transamination will form the glutamate, then glutamate to glutamine, then glutamine is released in blood and it is taken up by the liver where it will be converted, releasing the ammonia to form urea. So question number 25. The primary cause of the stone given in the figure is, this is cystinuria. I told you a cystinuria will result in the cystine stones. Cystine stones are hexagonal stones and these are cystine stones which you can find in the cystinuria. And what are the 4 amino acids which are lost in urine? Cysteine, ornithine, lysine, arginine. With this, we have covered your few questions on protein metabolism.